Hello everyone, today I would like to present the graph-based representation learning for spatial cellular communications. First of all, this slide briefly shows the timeline of the most important technology development with a special focus on the omics technology, starting from the bio-omics to the popular single-cell omics and to the recent spark-based spatial omics data, and then the recent emerging single-cell spatial omics data in 2021. And the single-cell spatial omics data has attracted great attention as it provides both the single-cell level and the spatial level insight into the cellular activities and cellular dynamics in complex tissues. Currently, there are three major technologies for the single-cell spatial omics data. And the major one is the nanostring cosmic spatial molecular image that starts in November 2021, and it is able to profile up to 1,000 genes, utilize the phage chemistry for identifying the RNA targets. The Visium scope starts in March 2020, and it is able to profile up to 500 genes. They utilize the morphage technology for high-throughput RNA readouts. And the Qianqian started in October 2020 and provides up to 300 genes, and they utilize the probe-based strategy to identify the RNA targets in the FFPE tissue slice. Here is the example that released by the different technologies, and the left figure shows the spatial slice from the nanostring. And this is the lung cancer dataset that covers 980 genes. Totally on this tissue slice, there are about 83,000 cells. And in the middle slice, this is profiled by the Visium scope that is from the liver cancer tissue slice. And this dataset covers 483 genes. And totally, there are about 600,000 cells on this tissue slice. And the Qianqian also released a breast cancer dataset that covers 313 genes. And there are about 167,000 cells on this tissue slice. So this different technology has provided us great opportunity to look into the spatial location of each cell, the tumor microenvironment. And however, as we mentioned above, that we are aware the gene expression has not very sufficient, and also the number of genes is not sufficient too, which causes great challenge for identifying the spatial cellular communications. And to combat this data challenge, we have proposed a novel method named as SPARCY to decipher the spatial cellular communications through the adaptive graph model, which is a tailored method for the single-cell spatial transcriptomics data, and this method has been published early this year in Briefings in Bioinformatics. And as shown in this figure, limited by the spatial data quality, the expression of ligand R can be very weak. So the true relations between the ligand R and its receptor R between the two adjacent cells cannot be detected directly. So the SPARCY model utilizes the gene-gene interaction patterns, including both the interaction pairs and non-interaction pairs, as well as the cell spatial graph, to learn a latent representation of genes, so that the true ligand-receptor interactions as well as the co-expressed genes are approximate to each other in this latent space. And in this way, the SPARCY model is able to predict the ligand-receptor interactions and other gene-gene association, including the transcription factors. Our SPARCY model projects genes into a latent representation through two components. One is the gene-based linear encoder, the other is the cell-based attentive graph encoder. Here, both gene expression patterns and the spatial relations between cells are incorporated into the latent space. The model is then trained through the triplet loss learning. Here, we assemble the positive anchor negative gene triplet from the interaction and non-interaction pairs. Such triplet loss defines a latent representation for each triplet, which assure a clear separation of the interaction and non-interaction pairs. And in this way, SPARCY learns the accurate latent representation of genes where the similarity between the interaction pairs is higher than that of the non-interaction pairs, and thus to infer if the ligand receptor pair has interaction or not. Our SPARCY model has demonstrated superior performance through benchmarking with current available methods, and here we skip this evaluation part that we would like to show the ligand receptor signaling that are identified by SPARCY, however, but missed by the other methods. Here we use spatial data from clonal sample, and for example, the learned latent representation, that is the embeddings of the ligand CD14 and ITGB2, were observed with strong associations in the latent representation. In contrast, both the CD14 and the ITGB2, they present extensive zeros in their raw expressions, with only a small portion of expressed cells observed, which explains that the low correlation of the CD14 and ITGB2 in the raw gene expression data. The other ligand receptor pair, the SAM3 and ITGB2, were also observed with strong associations in the latent representation but not in the raw gene expressions. These results demonstrate that the sparsity revealed the ligand receptor interactions, which otherwise could be hidden due to the data sparsity, which also explained why these ligand receptor interactions were hidden in their expressions. Moreover, we looked into the spatial expression of the ligand receptor in the local tissue region, and the blue dots represent the CD8 T cells expressing the SAM3, and the red dots represent the macrophage expressing the ITGB2. And those cells present very prevalent adjacent cellular interactions, which indicates that this ligand receptor does occur between the CD8 T cell and macrophage that are identified by the sparsity but missed by the other methods, demonstrate the accuracy of sparsity over the competitors for the single-cell spatial data. And then we applied to the sparsity to the nanostring dataset profile from a lung cancer patient with a total of 80 cells and 18 different cell types. And on this tissue slice, each dot should represent a cell, and each color represents a type of cell. The cell-cell communications identified by sparsity among the different cell types were shown in this code diagram, and the color-wise between the two cell types indicate their interaction strengths. Of note, we observed strong interactions from fibroblast endocellular cells and CD4 memory T cells to tumor cells, as well as the intra-tumor interactions. To quantitatively measure the interactions that occur for each cell type, we used a scatter plot to visualize the involved number and strength of ligand receptor interactions, where the x-axis represents the number of interactions, while the y-axis represents the interaction strength. This scatter plot shows that the fibroblasts and tumor cells are the most communicated cell types in cellular crosstalk. To investigate if the globally dominant crosstalk also exists locally, we scrutinized a specific region with abundant fibroblasts and tumor cells. Based on this local region, sparsity identified the cellular communications among major cell types. Consistent with the cell-cell interactions identified globally, we also observed strong interactions from fibroblasts to tumor cells. The involved ligand receptor pairs that contribute to the fibroblast-tumor crosstalk were present, along with the ligand receptor interactions from the major cell types to tumor cells. The gradient colors represent the interaction Specifically, the fibroblast expressed DCN and showed strong interactions with tumor cell expressed MET. Other ligand receptor pairs, including the DCN-MET, THBS1, ITGB1, and the collagen 18A1, ITG3, also contributed to the communications between fibroblasts and tumor cells. Moreover, the spatial locations of ligand DCN were located very adjacent to the receptor MET, and such phenomenon is also observed for the ligand receptor interaction of the THBS1, ITGB1, as well as the intra-tumor interaction between the collagen 18A1 and ITGA3, indicating that sparsity identified the novel discoveries and also the novel ligand receptor interactions between the fibroblast and tumor cells. Interrogation of the cellular crosstalk without elucidate the upstream mediators were not sufficient to fully understand the underlying mechanisms. So given the merit of our model, sparsity enabled us to predict not only the ligand receptor interactions, but also the interactions of any gene pairs. Therefore, sparsity was able to infer the potential upstream transcription factor of each ligand or receptor to facilitate the discovery of ligand receptor signaling activities. To achieve this, we collected the potential transcription factor of those tumor receptors and then sparsely reveal the most possible upstream transcription factor of receptors within tumor cells that mediate the ligand receptor interactions between the fibroblasts and tumor cells. Here, the second diagram presents the identified ligand, receptor, and transcription factor signaling axis with the source, that is the fibroblast expressing the ligands, and the receiver, that is the tumor cells, expressing the receptors that were mediated by the upstream transcription factors in the tumor cells. And different colors represent the transcription factor mediated ligand receptor interactions. From the second plot, we observed prevalent interactions between the THBS1 and ITTB1 with upstream transcription factors including the SMAD3, JU, and EST1. Next, we also evaluate the prognostic effects of these transcription factor-mediated receptors in lung cancer patients. Among the identified TF receptor pairs, patients with high SMAD3 ITTB1 expression has significantly poor overall survival in both adenocarcinoma and squamous carcinoma patients in TCGA, indicate that such transcription factor-mediated interaction axes may play important roles in lung cancer prognosis. So in summary, SPARCE is a novel adaptive graph attention model to incorporate both the spatial locations and the gene expressions of cells for revealing the active ligand receptor signaling axis across the neighboring cells. And SPARCE identified the upstream transcription factor mediating the identified ligand receptor interactions, which allows gaining further insights into the underlying cellular communications. And SPARCE is developed tailored for the spatial transcriptomics data and provided available as a ready-to-use open-source software, which demonstrates the high accuracy and robust performance over existing methods. And in this work, I would like to acknowledge our team, including Dr. Ziyang Tang, who just graduated from the Purdue University, Dr. Yang and Dr. Su from Purdue University and Indiana University, and also Dr. Wei Zhang from Wake Forest. And I would also like to acknowledge my funding support, including the Wake Forest Comprehensive Cancer Center Support Grant, and the Collaborative Research Pilot and the ACS Institutional Research Grant Pilot, as well as our Bioinformatics Shared Resource Course support. And thank you very much.